Hello everyone, myself Chandan, Assistant Professor, Department of Pharmacology, Sri Adi Chunchanagiri College of Pharmacy. So, today let us discuss regarding the cell and cell cycle in a clear and easy understanding manner. Now, moving on to cell. Cell is a basic structural and functional unit of all forms of life. Every cell consists of cytoplasm enclosed within a membrane and contains many macromolecules such as proteins, DNA, and RNA, as well as many small molecules of nutrients and metabolites. The word cell is derived from the Latin word cellula, which makes up the meaning small room. Cells can acquire specific function and carry out various tasks within the cell, such as replication, DNA repair, protein synthesis, and motility. Cells are capable of specialization and mobility within the cell. Most plant and animal cells are only visible under a light microscope with dimensions between 1 and 100 micrometers. Electron microscopy gives a much higher resolution, showing greatly detailed cell structure. Organisms can be classified as unicellular or multicellular. Most multicellular organisms are classified as microorganisms. The study of cells and how they work has led to many other studies in related to areas of biology, including discovery of DNA, cancer systems, aging, and developmental biology. A cell consists of plasma membrane enclosing a number of organelles suspended in a watery fluid called cytosol. Organelles literally means small organs have individual and highly specialized functions and are often enclosed in their own membrane. Let us see what are the cell organelles present in a cell. We have endoplasmic reticulum, which is subdivided into smooth and rough endoplasmic reticulum. We have Golgi apparatus, we have lysosomes, mitochondria, ribosomes, nucleus, and vacuoles. Now, coming to the function associated with the properties associated with plasma membrane. Plasma membrane consists of two layers of phospholipids with proteins and sugars embedded in them. The phospholipids molecules have a head which is electrically charged and hydrophilic, which means it is water loving, and tail which has no charge and is hydrophobic, which means water hating. These differences influence the transfer of substances across the membrane. Based upon the polarity, the substances or the molecules can diffuse in and out of the cell membrane or plasma membrane. These plasma membrane are made up of proteins, and it also includes membrane proteins which extends all the way through the membrane. They provide channels that allow the passage of substances. They also perform several functions, including giving the cell its immunological identity. They act as receptors for hormones and other chemical messengers. Some are enzymes, and as we already discussed, they provide a channel so that the ions or the substances can pass through. Now, coming to nucleus, the nucleus is the largest organelle and is contained within the nuclear envelope. It has a membrane similar to that of plasma membrane, but here it has tiny pores where the substances can pass between it and the cytoplasm. The nucleus contains body's genetic material in the form of DNA. This directs all its metabolic activities. Within the nucleus, it contains nucleolus, which is a roughly spherical structure which is involved in the synthesis and assembly of the components of ribosome. So we can say that it also regulates the production or the synthesis of proteins. Now, coming to mitochondria, These are membranous, sausage-shaped structure which are present in the cytoplasm, which provides ATP or which provides the energy for the cell by aerobic respiration. Ribosomes, these are the tiny granules composed of RNA and protein. They synthesize proteins from amino acid using RNA as the template when present in free units or in small clusters in the cytoplasm. Ribosomes are also found on the outer surface of the nuclear envelope and rough endoplasmic reticulum, where they manufacture proteins for the export of the cell. So, whatever the ribosomes which are present on the surface of the envelope, so nuclear envelope, and on the surface of the endoplasmic reticulum, they synthesize protein just for the sake of exporting it. So, Golgi apparatus packs it, and membrane-bound vesicles will transfer the particular proteins to the membrane, where, in the form of exocytosis, those synthesized proteins will be released out of the cell. Coming to the endoplasmic reticulum, endoplasmic reticulum is an extensive series of interconnecting membranous channels which is present in the cytoplasm. There are two types, rough and smooth. And how we are going to differentiate between rough and smooth? Here the ribosomes which are attached on the surface of the endoplasmic reticulum, they are known as rough endoplasmic reticulum. In smooth endoplasmic reticulum, they synthesize lipids and steroidal hormones. These are a set of synthesis of proteins, some of which are exported from the cell, that is enzymes and hormones that leave the parent cell by exocytosis. So these functions are associated with rough endoplasmic reticulum. Coming to Golgi apparatus, the Golgi apparatus consists of stacks of closely folded flattened membranous sacs. It is present in cells but is larger in that those synthesize and export proteins. The proteins move from endoplasmic reticulum to the Golgi apparatus where they are packed into membrane-bound vesicles. So by the mechanism of exocytosis, the proteins which are packed will exit the cell. Lysosomes are small membranous vesicles pinched off from the Golgi apparatus. They contain a variety of enzymes included in breaking down fragments of organelles and large molecules. Lysosomes in white blood cells contain enzymes that digest foreign materials such as microbes. Here we can give an example for neutrophils, where it is going to destroy particular microorganisms by the mechanism of phagocytosis. There the lysosomes play a very important role in destruction of that particular microorganism. Coming to cytoskeleton, this consists of an extensive network of tiny protein fibers. They are microfilaments. They provide structural support and maintain characteristic shape of the cell. Microtubules are larger contractile protein fibers that are involved in the movement of organelles within the cell, chromosomes during cell division, and in respect to cell extensions. Coming to centrosomes, this direct organization of microtubules within the cell, so it also plays a very important role in the movement of cell organelles. It consists of pair of centrioles and plays an important role in cell division. And we have cell extension. They include microvilli, cilia, and flagella. They project from the plasma membrane in some type of cells, and their main components of microtubules. See, these cell extensions play a very important role in the movement of cell organelles. Coming to cell cycle, many damaged, dead, and worn-out cells can be replaced by growth and division of other similar cells. At the end of their natural lifespan, aging cells are programmed to self-destruct and their components are removed by phagocytosis. Here, this particular process is known as apoptosis. Apoptosis is nothing but programmed cell death. This cell cycle occurs in two stages. mitosis and interphase. Mitosis was also known as M phase, and coming to interphase, this is the longer phase where it has three separate stages: G1, S phase, and G2 phase. In the first cap phase, the cell grows in size and volume. This is usually the longest phase. Here the cell prepares itself for cell division. Now coming to S phase, or also known as S phase, synthesis of DNA will occur in this particular phase. The chromosomes replicate, forming two identical copies of DNA. So at this phase, the individual cell has 92 chromosomes, means two pairs of genetic information or genetic materials will be available in a single cell. And coming to second gap phase, or also known as G2 phase, there the further growth and preparation for cell division will takes place. And there is another phase in interphase, means G0, which is nothing but resting phase. So if the cell division is not required at that particular moment, this particular cell will undergo resting phase. Coming to mitosis, we have prophase, metaphase, anaphase, telophase, and at last cytokinesis, where the cell divides into two identical cells. Okay, in prophase, what will happen? Here the chromatin becomes tightly coiled, means replicated chromatins will become tightly coiled. The mitotic apparatus appears. This consists of two centrioles separated by mitotic spindle. The centrioles migrate one to each end of the cell and nuclear envelope disappears. As you can see in the picture, you can see that centrioles are opposite to each other, and here the nuclear envelope will disappear. Coming to metaphase, the chromatids align on the center of the spindle attached by their centromere. In anaphase, the centromeres separate and one of each pairs of sister chromatids migrates to each end of the spindle as the microtubules that form the mitotic spindle contracts. So what are the mitotic apparatus which were usually present in prophase and in metaphase, how these chromatids were aligned at the center? Here the microtubules will contract. So what it does, it will separate this combined set into two different sets. So we have two sets of genetic materials in a single cell. In coming to telophase, the mitotic spindle disappears, the chromosomes uncoil, and the nuclear envelope reforms. As you can see in the picture, the nuclear envelope is beginning to reform. So we have here also we have two sets of genetic materials or chromatids. Now coming to cytokinesis, as you can see in the picture, the cell will divide itself in order to form two identical daughter cells. So they will be similar in function and they will be similar genetically. Thank you.